Welcome, everyone, to a special edition of the OHSU Informatics Research Conference. We normally meet on Thursdays, but at the end of the term, we have a number of students defending theses, proposals, etc. And so that's what we're doing today. We are hearing from Samantha Lawson, who's a Master of Science student in the Bioinformatics and Computational Biomedicine major. Samantha will be defending her thesis. And as most of you know, the process for that is that she presents her work, and then there are public questions. Then the public is dismissed, leaving behind her committee, who may ask her further questions, and then she is dismissed, and the committee decides whether or not the defense is successful. And of course, that's required to obtain the Master of Science degree in our program. Just a quick bio about Samantha. She received her Bachelor of Science in Biochemistry and Spanish from Whitworth University in Spokane. She's worked in a number of labs over the years, most recently serving as a research assistant at the Knight Cancer Institute for 3.5 years. She's currently the lab manager for Dr. George Thomas in the Knight Prostate Program, and her research interests include exploring genomic analysis tools to aid in the molecular biology environment. So I will turn the floor over to Samantha.
Thank you very much. Okay, let's do this. Okay. Thank you everyone for joining me today, and thank you Dr. Hirsch for the introduction. I'm just gonna kind of dive right in. So today I'm gonna start with the topic relevant vocab so that we all have kind of a baseline for knowledge base for the rest of the presentation. Move into our motivation, spend quite a bit of time on background information. Go through the research question and subsequent hypotheses, and then outline the specific aims. At that point, we're going to turn into what I've done since the proposal. So I proposed back in June, and since then, I'll talk about the methodology, results, discussion, potential future work, and then questions at the end. So my final working title for this thesis is Predictive Modeling of Ferroptosis Susceptibility Using Iron-Dependent Pathways in Renal Cell Carcinoma. I want to break this down into 4 key pieces so that we all kind of understand where we're going with the rest of the presentation. So predictive modeling, I emphasize this because I wanted to create a numerical tool that takes in one input measurement of a basal cell characterization, and then it gives that outcome variable of ferroptosis susceptibility. Ferroptosis susceptibility meaning how much inducing agent or how much treatment is needed to induce a cell type into ferroptosis, which is this newly discovered type of cell death that I'll discuss later on. And then I emphasized iron-dependent pathways. So this new mechanism has 2 separate arms, the lipid peroxidation side and the iron accumulation side. The lipid peroxidation arm of this, I feel, has been focused on more recently, and so I hope to kind of illuminate more of that iron accumulation side of this mechanism. I also believe that there's an interesting kind of research in metabolomics within the cancer research field. That's kind of a cool new topic. So I wanted to look into that. And then finally, renal cell carcinoma is the setting that I researched this in. So I built my model on those cell lines from different subtypes within kidney cancer. So the motivation for this project, 1, is to provide another kind of mechanism to target if treatment options of other mechanisms have proven ineffective and the tumor or set is resistant to those treatment options. So having this newer type of cell death creates a new target to look at. And so by adding more characterization to the knowledge base that's currently built on ferroptosis, you're kind of aiding that advancement in this mechanism. Next, I wanted to create an accessible assay-based method to model the ferroptosis prediction in these cell lines. And I want to highlight accessible here. The goal was to create something that the broader research community could use without too much time or cost expense. And so I was hoping to make a simplistic way for other researchers within this ferroptosis field to access and predict what will occur in their settings. Fourth, incorporate models into biological research with more frequency. So as I said earlier, kind of create this simpler illustration of what is going on within these cells. And then finally, give insight into how these different settings will behave in the hopes of transitioning into more heterogeneous contexts. Here we're starting in cell lines, but the idea would be to advance this into something more complex, such as an organoid or xenograft, or ideally into the human tumor tissue. A side note about this, ferroptosis can be used in multiple different ways. Since we are in the cancer context realm, the goal is to induce ferroptosis to kill those cancer cells. However, in neurodegenerative diseases, the goal there would be to inhibit those cells from going into ferroptosis to kind of conserve that brain cell function. And this is seen in Parkinson's, Alzheimer's, and it's even seen that with ferroptosis inhibiting treatments, they're able to lessen the damage from different stroke conditions such as ischemic and hemorrhagic. Okay, moving on to background. So as I said earlier, this is a fairly new type of cell death that was just named in 2012 by the Stockwell Laboratory in Columbia University in New York City. And that laboratory was kind of one of the key players in the early days of ferroptosis. So many of these early publications came out of that lab. They showed that when you add iron into a Avastin-treated, Avastin being a ferroptosis-inducing agent, which I'll talk about more, when you add iron into that setting, the cell death accelerates significantly. And then 2 years later, this mechanism was shown to be regulated by glutathione peroxidase 4. Glutathione peroxidase 4 is the main kind of enzyme in this mechanism, and I'll talk quite a bit more about it later on. A year later, lipid metabolism was shown to be involved in the mechanism. I wanted to highlight this mostly because, as I stated earlier, that this process is divided into those two parts, iron accumulation and lipid peroxidation. And just 5 years ago was that second arm identified, and so we can see the increase in how quickly this research is being advanced. And then 2 years later, PEs, or phosphatidylethanolamines, were showed to be oxidized during this process. Again, another example of how this is being narrowed down as PEs are a species within that lipid metabolome. Moving on to the actual process. Now, I know there's a lot going on in this slide, and so I'm just going to highlight a few things. Panel A is what I'm going to focus on. So we all have kind of some background knowledge of what's going on within this process. However, Panel B is this iron metabolism side. Here I just want to illustrate the complexity of what goes on in this arm of the pathway. So to begin, we're going to start with our Fe2+ over here, or our iron pool. This undergoes a Fenton reaction, which the iron combines with hydrogen peroxide and creates toxic free radicals within the cell. Now, over here we have our GPX4 regulator. This is able to take glutathione, react with these lipid peroxides so that our cell does not go into ferroptosis. If the GPX4 is not functioning, that those lipid peroxides accumulate within the cell and kill the cell. So another point, another targetable area is this cysteine glutamate antiporter. This brings cysteine within the cell, which is then converted to glutathione, which GPX4 then uses in that interaction I just mentioned, where it is able to use that lipid peroxide glutathione and remove it from the cell. Two notes here. This is a regulated type of cell death. It is not accidental, and it can be distinguished between the other types of cell death, such as apoptosis, necroptosis, etc. It can be that is done by using ferrostatin here, which, if added to a ferroptotic cell, will rescue the cell. If added to any of the other, like apoptotic cell, it will not rescue the cell and it will undergo that cell death halfway. So these, again, the cysteine glutamate antiporter and GPX4 are targetable areas for inducing ferroptosis. Another note is over here, this kind of section is the iron chelator. So a DFO is an iron chelator that mops up that excess iron within the cell and will inhibit the cell from undergoing ferroptosis. Okay. So we have our targetable areas. I want to talk about the inducing agents now. A small molecule assessment was done to determine which compounds were able to induce cells into ferroptosis. Multiple compounds were found, and for my purposes, I want to focus on arrestin and RSL3. Arrestin is a class 1 ferroptosis-inducing agent, meaning that it inhibits that cysteine glutamate antiporter. Cysteine cannot come into the cell, cannot go through that interaction, and then use glutathione to combine with lipid peroxides. RSL3 is a class 2 ferroptosis-inducing agent. This inhibits the GPX4 system to induce the cell into ferroptosis. So we have multiple targets, and with that, we're able to understand or kind of manipulate the mechanism at two different parts. There are other inducing agents that have been shown to be successful. However, they do also target those same areas. Okay, moving on to drug sensitivity. So for our outcome variable of this model is drug sensitivity, which will tell us how susceptible our cells are to ferroptosis. So because it's our outcome variable, I wanted to ensure that it's the most consistent and accurate metric for this model. So after some research into drug sensitivity metrics, it was determined that IC50, or the normal or traditional type of metric for drug sensitivity, is correlated with seeding number, meaning that the experiment conditions heavily influence this metric. IC50 is the the inhibitory concentrate at 50%, meaning that with our control cells, it's the dose at which our cell count reaches 50% compared to our control cells. So growth rate, GR50, which takes into account that seeding number or those experimental conditions, looks at what the number of cells that were seeded, our control cell count, what rate of growth that was, and then the dose at which that rate of growth is 50%. So because this is shown to be more consistent and it's not influenced by those seeding numbers or experimental changes, that is what I decided to use as our outcome variable for this model. And then moving on to renal cell carcinoma. So renal cell carcinoma is one of the top 10 most lethal cancers in the US and is most sensitive to ferroptosis. Over here we show how different cancers react to a class 1 inducing ferroptosis inducing agent. And here we can see that renal cancer is affected the most. But we also can see that there's a wide range of sensitivity across these settings. And so over in the more shallower graphs, you can see that lessen sensitivity. So the biology behind renal cell carcinoma and why that is more sensitive to ferroptosis has been shown recently. And it has to do with the majority of renal cell carcinoma is made up of a clear cell subtype, about 70%. And that undergoes a mutation quite frequently within the VHL or von Hippel-Lindau tumor suppressor, which then is inactivate. Down the path leads to an activation of this hypoxia-inducible factor pathway, which leads to an increase in angiogenesis and those hallmarks of cancer. And another kind of an output of this is an increase in the polyunsaturated fatty acids of the cell. It creates an enriched state of those lipids that I mentioned earlier. And the higher accumulation of these lipids at the basal state before any treatment is introduced leads to a higher sensitivity to ferroptosis. So that made kind of led to the conclusion that those PUFAs, or those polyunsaturated fatty acids, are the rate limiting agent in this mechanism. Okay. I'm going to explain some assays that helped inform the model. So here we're looking at immunofluorescence, or sorry, just fluorescence images of labile iron pool within the cell at different localizations. So as I said, that iron accumulation side of this mechanism is what I'm focusing on. And so this is one of the features that I wanted to look at for the model. This is somewhat new chemistry. It's only been, the publications that have used this assay have only been around for, or published within the last 5 to 6 years. And there's multiple localizations, as you can see across the top here. These are all different assays. So to choose what would be most accurate and consistent for my model, I took into account that here you're looking at control cells within the mitochondria. So we're going to focus on this area here. And we see that our iron signal is low, and it's increased with a ferroptosis-inducing agent. Right? That's what we would expect to see. However, when you induce ferroptosis and you add on that iron chelator that I mentioned earlier, you should see a decrease in iron as that iron chelator is mopping up that excess iron. And you don't see that here. So I stayed away from this assay. And similar here, you see the same trend, but it's not as significant as in the endoplasmic reticulum and Golgi localization of the labile iron pool within the cell. And so this is all quantified in this bar chart over here. You can see that the trend is most significant in this localization. And the authors of this paper did this at a time point, which showed at other times this assay here was the most consistent. So after taking this into account and looking at manufacturers' recommendations, I chose that Ferronox 1, which is this assay here, is the most consistent and accurate for measuring that labile iron pool. However, other assays have their specializations. So LisoRonox measures this localization. And then there's Ferrofar Red, which also measures the ER, but is used for more flow cytometry purposes. Okay, bringing this into a little more database, so FairDB was published in early this year, March of this year. It's a database specific to those markers, characterizations, diseases, and agents for ferroptosis, and they also have other functionalities to visualize the networks within this mechanism, and it is what I used to pull, among other things, to pull features for my model. I think it's a kind of a great environment for ferroptosis researchers to contribute, and they're able to kind of add to this knowledge base. This was done by a group out of Guangzhou and Chengdu, China, and it still is growing very rapidly. I also believe that with added functionality, such as a model that is created for the research community to use, would be a good opportunity to kind of contribute and bring all of the researchers together to help kind of advance that field there. So, modeling options. So there have been a lot of models created for other types of cell death, apoptosis, necroptosis, etc. For ferroptosis, not so much. This was one that was published this year. It is a more simulation-based model, takes in quite a few different features, and it's granular. It's a 0, 1, 2 bin, and there are intermediates along the way, and it shows what should occur within the cell. It's predictive in the fact that they were able to test this on a ovarian stem cell line and confirm that this in fact did occur. And so this has helped inform the model that I wanted to create. However, it was a much different motivation. Again, I created something that is more, I'd like to say, somewhat accessible, so that we have one feature going out in and we're able to predict how the cell will react. Okay, so our research question: can iron-dependent probes, including protein abundance and assay measurements, predict ferroptosis sensitivity in a solid tumor cell line? And if that is possible, can this be validated and generalized across contexts to other cancer types? And again, this is looking at those innate factors within the cell. We're using those basal measurements. Our hypothesis is yes. These markers can be utilized as direct predictors for susceptibility, and once that is quantified, they can be expanded to other contexts. So our specific aims: identify the predictors that inform ferroptosis cell fate. And so again, these were pulled from FairDB and other previous publications. And I want to note that I'm purposefully leaving out the iron-dependent probe specification here, because I want to pull all features that could be a predictor in our model. In case we determine that no, iron-dependent probes are unable to predict ferroptosis successfully, I would still like to create a model that is based off other types of probes, such as oxidative stress and the lipid peroxidation that we chatted about. And then, 2, develop a mathematical model to predict ferroptosis susceptibility. And again, emphasis on this mathematical prediction, so that we're not using a granular system. We're trying to create a more fine-tuned prediction method. And then determine the extent at which this model can be generalized. Kind of see if we can apply it to a different context, and if we can't apply it, where are those limitations? So, new stuff, the methodology that in the last 6 months I've put together. So I started with viability assays using cell titer blue. This was done over 72 hours, and this again is our outcome variable. This shows how sensitive each cell line is to our ferroptosis-inducing agents, Avastin or RSL3. As I said earlier, priority was given to iron-dependent assays. However, I did pull in anti-malondialdehyde as an antibody to measure ferroptosis, to measure that oxidative stress of the cell at the basal level to see if that can inform our model. This was a 48-hour incubation and was done via IF. Protein abundance was quantified using Western blots, and this was the specific markers I were looking at was HMOX or heme oxygenase, DMT1, a divalent metal transporter, or FTH, ferritin heavy chain. So again, these are iron-dependent probes that we were looking at. And lastly, iron assays, ferronox1 and ferrofar-red that I spoke about earlier. Quantification was done using ImageJ as it's the most accessible, popular, open source tool that's used for quantification in these settings. And then data organization and visualization was done in R. There were quite a few packages that I'm very grateful for that were published in the last couple months, actually, that helped with visualizing those drug sensitivity metrics and some of our model specifications. So, classic model building steps here. However, we're going to spend most of our time on feature selection. So, earlier I pointed out the IF quantification and the Western blot quantification. Our anti-MDA signal was great. It was consistent, good quality data. I did exploratory data analysis on all of our features, all of the possible features, and then looked at correlation analyses as well. So anti-MDA held up to the standards there. However, it's not an iron-dependent probe, and the biology behind why that marker at a basal level would induce ferroptosis is a bit more unclear at this point in time. Quantification of Western blots was performed using Image Studio, which is one of the most popular quantification methods. And after EDA and correlation, actually we didn't get to correlation analyses, after EDA showed that there was no consistency between assays, it did not hold up to standards. And after a bit of research, this has been shown in multiple publications. There is one in 2018 that compared all of these different quantification techniques of Western blots and showed that they are misleading in nature due to a discrepancy between amount of protein being loaded. But that's a topic for another day. And so after all of that, after discovering which features were one consistent, good quality, and then looking at correlation analyses with our outcome variable, I decided to stick with our labile iron pool measurement that I discussed earlier. And then we moved on to model selection with one feature and a good correlation, simple linear regression, and then it was tested and validated on other cell lines, which I will talk about shortly. Okay, so here are our outcome variables. It's a bit confusing, but I'll walk through. So each line here is a different cell line. This is measuring that growth rate metric that I was discussing. So we can see between here that the lines aren't identical between treatments, but they are similar. And so we know that we are hitting different targets within that pathway. So from these curves, we are able to calculate that dose at which 50% of the growth rate occurs. And so from there, we were able to correlate with our chosen feature selection. Here is that first iron measurement that I was talking about. These are ferronox1 assay measurements or images. So ferronox1 attaches to that mitochondrial labile iron pool. And we can see the signal here. So with our TGA10, which is the most resistant cell type, we see a very limited iron signal. Move over to ACHN, which is a more sensitive cell type, and we see an increase in iron signal. And then finally, on our right here, we see this ACHN cell line with the GPX4 gene knocked out, meaning that this requires no treatment to be induced into ferroptosis. So I used this cell line, this genetically modified cell line, as an extreme and a control. All of our cell line iron signals measured below this, and I will show you where that would lie in our correlation analyses. So, see a direct correlation between the iron signal and drug sensitivity. If we require more RSL3 dosage, we have a lower iron signal. So if we need a higher dose of RSL3 to be pushed into ferroptosis, our iron signal will be at its lowest. If we need a small amount, if we're very sensitive to RSL3, then we will have a high iron signal. If we mapped that knockout cell line onto this plot, it would be way over here. We have, it needs 0 to be induced into ferroptosis, and it has extremely high iron signal, the same with Avastin. And then, because I was able to use only one feature, making the model more accessible, I wanted to ensure that it was accurate and stood up to a secondary assay. So I performed ferro-far-red, which also measures that mitochondrial localization. And here is that iron signal for the resistant cell type, for the sensitive cell type, and then again, the extreme control of our GPX4 knockout cell line. So, specifications for this linear regression, we're able to account for more variance in our RSL3 model than in our Avastin model. It's still, and I'd like to kind of point out why this is. So we have to take into account the dosing range for each inducing agent, because if that range is greater, we're going to see more variability within our data. RSL3 dosing range is between 5 nanomolar and 100 nanomolar to see those cells be induced into cell death. Avastin is around 50 nanomolar to 5,000 nanomolar. And so we'll see a variation within the data showing that the cells react more, the cells react in a more predictable way when the dosing range is smaller. Our p-values show this is significant, and so we're happy with it. So, on to the last specific aims. I chose to try to generalize this to V160 cell line, which is a prostate cancer cell line, and took the cell viability and the ferronox-1 iron signal and tried to apply this model to a separate context. Here we see very shallow curves for our inducing agent. And we also see a very low iron signal. So this fits with our trend, meaning this cell line is very resistant to ferroptosis, and it has a minimal iron signal to ferronox. So try to apply our model. However, our cell viability here has a GR50 of infinity. And so we are unable to accurately apply this. It's, I believe it's a good example of it fitting our trend. However, it also illustrates the boundaries that this model has. If we build it on the most sensitive cell type and cancer context and try to apply one of the least sensitive cell lines or cancer contexts, it's going to show these boundaries. And so narrowing that gap between the most sensitive and the most resistant could be helpful in expanding this model. Okay, discussion. So, one, we were able to find significant and reliable features. So again, the iron measurement within the cell showed that it could be consistent across. It was quality data. We have that single feature input, which makes it accessible to other researchers. And it's one of the shortest protocols I looked at for the features to consider. 2A, linear regression model was created to predict how the cells would undergo ferroptosis with a certain amount of inducing agent. And then the model proved to be not generalizable, but with more work as to generalize it to more sensitive cell types would be useful in this setting. So some future work. Again, continue with those iron measurements as they proved effective and consistent. And then also look at that anti-MDA feature that I talked about. Again, good data, but the biology behind it needs to be explored more to make it useful in this context. As this is a somewhat newer type of cell death, the pathways are still being determined and characterizations are still being made. So we may find other targetable areas or other inducing agents as this work is being done. As I said earlier, use a wider range of cancer contexts so that you're able to kind of build this model gradually up instead of kind of jumping from point A to point B. And then share in a collaborative space, such as FairDB. Create functionality in these kind of contributive resource hubs so that other researchers can also add and find helpfulness in those computational tools. So I believe that is it. Are there any questions?
Great. Thank you for your talk. And we will open the floor to questions. People can either unmute and ask, or they can type into the chat box. Okay, well, I'm sure that your committee will have some questions for you. So at this point, I want to thank Samantha for an interesting talk, and we'll dismiss everyone. We'll ask the committee to stay, and we'll ask her any further questions. So thank you.
Thank you.
Mhm.